Hey everyone, so I'm just going to take a moment to go over the cell cycle. So this is what you saw last class in the video, and then the interactive click and learn about cancer. So just to elaborate a little bit and make sure we're all on the same page, I want to go through step by step. So we're going to start with the longest part of the cell cycle. The longest part of the cell cycle is called interphase, and it includes all three of these pieces here, okay? So this entire thing is called interphase. This is where cells spend most of their time. This is where they do their cellular functions, right? This is where a liver cell carries out the functions of a liver cell. This is where a brain cell carries out the functions of a brain cell. This is their day-to-day, okay? And then it gets broken down a little bit further. So this first phase, the first part of interphase, is called G1. And in G1, the cell grows. I mean, that's as basic as it gets. It duplicates everything except for its DNA. It copies organelles, and it just gets bigger, okay? The cell has to grow in size. Let me show you why. So the cell is going to be dividing, right? Now, if you took this cell and split it in half, you'd have a cell this big and then a cell this big. If you took this and split it in half, you'd have a cell this big and then a cell this big. So if you split a cell directly in half, it's going to shrink every time. But if you take that cell, grow that cell first, then split it in half, it winds up being the same size. So that is what G1 is for. The cell has to grow, and it has to also copy everything except for the DNA. The DNA gets copied in S phase. This is where DNA replication occurs. That whole thing we learned about last week with helicase and DNA polymerase and all of that comes into play during S phase. So this is where DNA replication occurs. The S actually stands for synthesis of new DNA. So everything we learned about that semi-conservative replication, where the DNA molecule, which is bundled up into a chromosome, gets opened up, and each strand gets copied so that you wind up with two identical copies of DNA. You don't want any mistakes here, because the whole point is that you're trying to make a new cell, and that new cell can't have any mistakes in its DNA, okay? The next part of interphase is called G2, and this is, I mean, depending on what type of cell it is, basically what textbook you're looking at, G2 does different things. For us, it's going to be where proteins and organelles are copied, okay? And basically the cell just prepares for division, okay? Now remember that there are checkpoints throughout this entire thing, okay? And the checkpoints serve to regulate cell division. So if the cell is not ready to divide, those checkpoints will stop it from dividing. And it's when these checkpoints don't work anymore, when something gets mutated, that cancer can occur, okay? So there is a checkpoint right after G1. There's a checkpoint right after G2. And then there's also a checkpoint in the middle of oops, that's not what I meant to write here. Oops. Checkpoint. And then another checkpoint. So all of these checkpoints serve to determine whether the cell is healthy, whether there are any mutations, whether everything is ready to go, okay? This checkpoint is really, really vital, this one right here. It says, does this cell have everything it needs to divide correctly before this whole section here, okay? This one's also important. It says, you know, should we be replicating our DNA? Are we ready to replicate DNA? Because that's really, I mean, if the cell has 2 copies of each chromosome, then it is going to be dividing, right? So these checkpoints are in place to regulate cell growth and cell division. If these checkpoints get messed up somehow, if some protein or some enzyme gets mutated, then the cell can divide unchecked. It divides uncontrollably, and there's no regulation, and that's when cancer can occur. This is why tumors grow, because it's unregulated cell growth. It basically becomes a lump of cells, okay? So then this whole thing here is M phase, okay? I'll make M phase different color. Okay, and M phase is further broken down into 2 parts. These 4 parts here are called mitosis, and this part right here is called cytokinesis. Okay, so the first part of mitosis is prophase, the second is metaphase, the third is anaphase, the fourth is telophase. And you'll learn more about these as we go on. For right now, just know that there are 4 parts of mitosis. Mitosis is defined as the division of the nucleus. So everything in the nucleus gets duplicated in S phase of interphase, right? That's where DNA replication occurs. So mitosis really only serves to get those doubled up chromosomes separated. It's the separation of the nucleus. Cytokinesis is the division of the cytoplasm. So in mitosis, it's just the division of the nucleus. This phase right here is where you finally get 2 separate cells. Started as 1 cell here growing, cell cycle ends in 2 identical daughter cells, okay? 2 identical daughter cells by the end of cytokinesis. And then each of those daughter cells will start their own G1. Each one will go into G1 if they need to divide again. If they do not need to divide again, they will exit the cell cycle okay? So if the cell does not need to divide anymore, let's say it's a neuron. Neurons don't really divide. You can't really make new brain cells. Once you lose them, you lose them. So a lot of them enter G0 so that they stop dividing. However, if they need to divide continuously, the new cells made after cytokinesis will each enter G1 again. So it is a cycle that is continuous until, number 1, the cell enters G0 and stops dividing, or the cell dies. All right, so this is the cell cycle. In the upcoming classes, we'll prepare for learning about mitosis. There's a lot of vocab that we have to learn, which you'll actually see today. And then next time we'll learn about each of these steps: prophase, metaphase, anaphase, and telophase. All right, thanks for tuning in.